En un momento comenzamos. Bueno,
Está bien, sí, sí.
ya estamos en vivo.
Ok, muy bien. Pues voy a hacer la introducción, la presentación. Sí, ya se puede, ¿verdad? Ok. ¿Qué tal? Buenas tardes a todos. Gracias por estar en el seminario, en el coloquio del día de hoy, perdón. Antes de empezar la plática, nada más me gustaría decirles que, por favor, escriban sus preguntas en el chat del YouTube y yo leeré las preguntas al doctor Chanda para que él las responda. También ha dicho de que si hay algunas aclaraciones que ustedes deseen hacer durante la presentación, pues lo pueden interrumpir y si no, pues él aclarará todas sus preguntas que tengan al final del seminario. Today, it is my pleasure to introduce Dr. Baron Chanda, Professor of Anesthesiology at the School of Medicine of Washington University. Dr. Chanda is a native from India. There, he studied biochemistry at the University of Delhi and biotechnology with a specialization in biophysics at the University of Pune. Then, he obtained his PhD at the National Center for Biological Sciences in India, studying membrane proteins. In 2000, he joins Dr. Besanilla's lab at the University of California as a postdoctoral fellow, where he gained expertise in the gating mechanism of sodium and potassium channels. He joined the Department of Neuroscience at the University of Wisconsin-Madison in 2006 as an assistant professor. In 2020, he moved to Washington University School of Medicine as a professor of anesthesiology. Dr. Chanda's efforts are directed to gain a deep understanding of ion channel function in a physiological context. He has made significant contribution to understand the thermodynamics and voltage sensing mechanism of ion channels. Today, Dr. Chanda will present the seminar entitled Proving Allostery in Ion Channel at Single Molecule Resolution. Dear Baron, welcome to San Luis in a virtual manner. Hopefully one of these days you can visit us in in person. Go ahead, please.
Okay, thank you, Jorge, for this opportunity. I'm going to start with sharing my screen. Let's share. Okay, can you see my main presentation slide?
Yes.
Okay, good. All right. So thank you, Jorge, for this opportunity and to share my research, and thanks for the kind introduction. So I will start with a topic that is very near and dear to my heart, allostery and thermodynamics of ion channel. And first, I'll just give a brief introduction before I dive into this. And as I mentioned to Jorge, you can, if you have a question that requires or you want a clarification, feel free to interrupt me. If it requires a more detailed answer, I will push that question back to the end of the seminar, and that's when I'll answer it. Okay. Okay, so you can see the change in slide, correct? The new slide, right?
Yep.
Okay. Yeah. So we work on pacemaker ion channels, and this work in my lab started about 10 years ago, before that, I used to work on voltage-gated sodium channels. And the reason these channels are interesting to us and NIH and to the general public as such is because they play a central role in many rhythmic circuits. For instance, in the human heart, they are found in the sinoatrial node, as shown here. I'm going to use a laser pointer, as shown here. And in that sinoatrial node, these channels contribute to what is known as the diastolic depolarization. So on here, you have the sinoatrial node action potentials, and you can see the action potentials come down due to opening of the potassium channels, which repolarize the membrane. But when they come down, they do not, at the resting membrane potential, they do not stay there. There's a depolarization that occurs. And this depolarizing current is due to opening of these pacemaker ion channels, or the HCN channels. Okay, HCN stands for hyperpolarization and cyclic nucleotide activated ion channels. So this depolarizing current then brings the membrane potential close to the threshold, which causes the next action potential to fire. So it turns out that many of these excitable cells, which have these self-propagating action potential, that the action potential keeps regenerating again and again without external stimulus, they have these pacemaker ion channels. And in the heart, these channels, for instance, also regulate the frequency of these pacemaking. So during fight-flight response, the adrenergic stimulation due to adrenaline in the bloodstream that activates second messenger pathways, and that increases the cyclic AMP concentration. And these HCN channels are also partly activated by cyclic AMP, which in turn makes these channels easier to open. And as you can see here, there are more currents, as shown in red, and that increases the frequency of these action potential. So this is what underlies a fight or flight response. So these channels are physiologically very important. And they are not just found in the heart, they are found also in the nervous system, they are found in the peripheral as well as in the peripheral nervous system and central nervous system. The mutations of these channels are associated with a variety of diseases, including epilepsy, neuropsychiatric disorders such as schizophrenia, autism spectrum disorders, depression. In addition, they have been implicated in Alzheimer's disease, Parkinson's, and others. And what I will say is that these channels are relatively understudied compared to what I used to study, like the voltage-gated sodium channels. The number of gene mutations that have been mapped to disease are still much lesser than what I know is out there for something like more established channels like sodium channels, potassium channels. So if you express these channels in cells or oocytes, one of the features of these channels that makes them stand out is that these channels open on hyperpolarization. So this is a typical activity curves that we use on the y-axis here. We have plotted the apparent open probability, and on the x-axis is the voltage. And what this shows is, as you hyperpolarize the membrane, the channel open probability goes up. That means the channel is more likely to open. And in presence of cyclic AMP, it becomes easier to open. That's this rightward shift of the open probability versus voltage curve. In addition, the fraction of open channels also increases the what we call as the maximum PO of the channel. So this is this cyclic AMP dependent regulation is isoform dependent. So HCN2 is much more dependent, much more sensitive to cyclic AMP, whereas isoform HCN1 is not as sensitive. It only, cyclic AMP causes only 5 millivolt shift in this PO versus voltage curve. And that's not because cyclic AMP doesn't bind to HCN1. It binds to both HCN1 or HCN2, but the effect on HCN1 is much less. And as I was mentioning in the previous slide, these channels are also activated by cyclic AMP. So these channels on their C-terminus end have a cyclic nucleotide binding domain, which is shown here, called CNBD. And as shown in this panel here, that increasing cyclic AMP concentration in the cytosol causes that to bind to the CNBD, which favors channel opening. So I think the thing that I would like to emphasize here that these channels, like BK channels, they are central in signaling pathways. They integrate both electrical and chemical signaling pathways. So my lab essentially works on two areas when it comes to these channels. So we are trying to understand how ligand binding activates these channels, and that these studies involve single molecule studies, which is what I'm going to talk about today. And the other part of my group is interested in using structure and functional methods to understand how these channels activate in response to voltage. Because their activation mechanisms are unique, because most voltage-gated ion channels activate on depolarization. And this is somewhat unique. HCN channels are unique in that respect. We want to understand what causes these channels to activate upon hyperpolarization. I'm not going to talk about this work today. What I'm going to focus on is a single molecule studies to understand ligand binding and allosteric. Okay, so here I'm showing you the structure of the HCN channel. So the transmembrane region structure is very similar to a typical voltage-gated potassium channel, sodium channel. Okay, what is different here is the cytoplasmic domain, this region here, which is the CNBD domain. So these channels are tetramers. Four subunits come together to form a HCN channel or a pacemaker ion channel, and each subunit has this cyclic nucleotide binding domain. So in total, there are 4 potential cyclic nucleotide binding sites, and there have been numerous studies in this trying to understand how cyclic nucleotide binding causes channel activation. And I won't have the time to go through all of that. What I'm going to show is what for our purposes was some of the most interesting findings that motivated some of this work. So on this side, this panel, you have this data which shows the binding, microscopic binding constant for each of the ligand that binds to cyclic nucleotide binding domain in HCN2 channels. So on x-axis here is the first ligand binding, as you can see, shows a certain binding affinity, but this binding of the second ligand is more favorable, whereas the binding of the third ligand is less favorable. Okay, and then again, the binding of the fourth ligand becomes more favorable. And this is, in fact, the ligand binding, it turns out, is also dependent on membrane voltage. So if the membrane voltage is minus 30 millivolt, you have one sort of cooperativity. But if the membrane voltage is minus 130 millivolt, you have another sort. In fact, this non-monotonic cooperativity is much more pronounced at more negative membrane voltages. Okay, and I should add that all of this work came out from work that was done more than 10, 15 years ago. And this was state-of-the-art measurements using patch clamp fluorometry, where they had measured direct ligand binding. And but these were all macroscopic binding measurements. That is, you are not doing single molecule, you're looking at binding of the a large number of protein HCN channels to a large number of cyclic AMP at the same time. All right, so one of the issues that is a limitation, and I think that is not appreciated as much of macroscopic binding, is this problem of parameter identifier. So what I've done here is taken a figure from this paper published by Rick Aldrich's lab. And in this figure, what they are showing is a model of calcium binding to calmodulin. And this is experimental data of the binding curve that was measured by various labs. So each of these different symbols shown here is data that were obtained from different labs. So there are about 4 or 5 different data sets. And as you can see, that there is a generally pretty good agreement of how the binding, what the binding curve looks like, right? And all of this data was fitted to a very simple model. All the groups fitted this data to a very simple model, which is a sequential model, okay? And which is there are 4 binding sites. K1 is the first ligand binding, K2 second, K3 third, and K4 fourth. So there's a sequential binding. And the idea was that what are these, each of these equilibrium constants correspond to? And it turns out that these different groups over the years published data that that showed that all sorts of cooperativity. In fact, you can also observe non-monotonic cooperativity by, that some groups have reported, whereas others report very little cooperativity, if or negative cooperativity, if any. So how is that possible? And this is what they defined it as a problem of parameter identifiability. And the reason this happens is that you have only one set of data, this binding curve, and you don't have, you cannot uniquely constrain each of these individual parameters, okay? And so essentially, you are trapped when you do these optimization, you are trapped into what would be called the local minima, and you get these various microscopic equilibrium constants. So how can one resolve this? And the way to resolve this is to measure each of these binding constants separately, right? So if we can measure binding of 1 ligand, 2 ligand, 3 ligand, or 4 ligands individually, then we would be able to resolve this from these measurements. And if you wonder that this is, whether this is how big of a problem it is, and you think about this going back all the way to hemoglobin, which is considered an exemplar for probing allosteric. You have 2 sorts of models, right? You have the MWC model and you have the KNF model. And in fact, even to date, if you just take the hemoglobin binding curve, both of these models fit pretty well to just the hemoglobin binding curve. What you really need is to determine how the distribution of the individual ligation states are changing with response to concentration. So if you were to get those distributions, then you can constrain and determine which is really the right model that fits or describes your data. And so this is really very hard to get, is to identify what is the concentration dependence of the individual ligation steps. And this is what we were aiming to do at a single molecule level. How does one do that? Okay, for instance, you consider this simple model where I have considered a molecule, a dimer, and there are two ligand binding sites. And for instance, what I've shown here is that each of the ligands have a different color. And if they are both independent, then I can look at ligand binding. So each of these bumps here, okay, or step jumps that you see corresponds to ligand binding. And if they are independent, then if one is bound, it has no effect on the binding of the other ligand, right? So the dwell time that you measured here, bound dwell times, is the same in this condition versus that condition. Whereas in this condition, there was another ligand bound, but it did not affect the binding of this ligand. So that's truly an independent mechanism. Whereas if it was a cooperative mechanism, then if one ligand is bound, it affects the dwell time of the other ligand. Okay, but if that is not present, it has a different dwell time. So that tells you the extent of cooperativity between these two processes. Okay, so that's kind of the simple goal. In our case, we don't have different colors for different ligands. We use the same ligand, but we can count the number of bound ligands by looking at the intensity jumps. And that's what we will do. Okay, so how can one do these experiments? One way to do this is in a typical single molecule experiment. What you do is you take a cover glass, you attach your receptor to the cover glass, you tether it down using various methods that are available, and then you take a freely diffusing fluorescent ligand and add it to the solution. And then a typical approach is called the total internal reflection microscopy, or TIRF. And in TIRF, what you do is you come in with an excitation light, and the excitation light bounces off the cover slip. But it also generates an evanescent field on the other side. And this evanescent field decays so that the excitation only is for the excitation volume is restricted to very close to the cover glass. So in this situation, all of these freely diffusing ligands which are in your solution are going to be dark, and the ones that are bound to the receptor, which is close to the glass surface, are going to light up. So you can use that as a way to detect ligand binding. So this works well as long as so this is just an example, and this works well as long as the fluorescent ligand concentration is less than 10 nanomolar. As soon as you start going to higher ligand concentration, okay, this doesn't work, because what happens is all these fluorescent ligands that are sitting out here, those also, there is enough concentration within this excitation volume, even though this is restricted, but as greater than 10 nanomolar concentration, there is enough excitation that you basically will not be able to resolve single ligand binding steps from all the fluorescent ligands that contribute to the background. And this is in the field is referred to as concentration barrier for ligand binding studies, okay? And for us, this is a problem because our KD is in the 1 micromolar range. So we cannot use this method. And in fact, it turns out that most ligand binding processes that involve a ligand binding that drives some sort of a gating process is in this micromolar range. So for most studies, this method of using TIRF will not work for studying ligand binding. I just said this, as this shows from the Brenda database, the Km for enzymes binding to substrates. And as you can see, most of the enzyme substrate, the Km is in the micromolar to millimolar concentration, and using TIRF, it's not accessible. Okay. So the way we get around this is this technique called zero mode waveguides. What it is essentially we take a cover slip and we coat it with aluminium. And then in this aluminium, we make holes that are less than the wavelength of light. So these are fabricated in using ion beam or electron lithography. But the holes are about 100 to 200 nanometer diameter. So what's the advantage of making a hole of this size? So the main advantage is if you now come with light from the other side and try to push the light through to the through the cover glass onto the other side, what happens is most of the light will not go through because of this aluminium coating on that, right? But that but it will create an evanescent field that is on the other side of the glass, with a very short length constant. And this evanescent field now will you are essentially reducing the excitation volume in this way. So this technique then allows us to reduce the excitation volume or the observation volume by an order of magnitude or 3 orders of magnitude. So in case of turf, as shown here, is 10 to the minus 17 per liter, but with ZMWs, you can reduce the excitation volume to what is called the zeptoliter, 10 to the minus 21 of a liter. And this reduction then enables us to have very high concentration of fluorescent ligands in solution. And this ligand, when it comes and binds to this receptor, then you can observe that ligand binding. And that's what this ZMW does. It allows us to access ligand binding or study ligand binding in the micromolar range. Okay. I put this slide up just for comparison. There are multiple techniques people have developed, and ZMWs, as it can, this is the concentration upper limit for most of the techniques, and here is for turf, and for ZMWs, it is 10 to 80 micromolar. That's the concentration limit. And for hybrid techniques, it's 100 to 1600 micromolar. So that this is also something that we have developed, ZMW plus FRED, and this allows us to even monitor ligand binding at millimolar concentration. Okay, so just a brief look at how these experiments are conducted, what is the setup that we use. But before I do that, I want to acknowledge the people who were involved in developing this. Marcel Goldschen-Ohm was a postdoc in my lab when he started this work, and now he has run his own lab at UT Austin. Randy Goldsmith is my long-term collaborator from UW Chemistry. David White was my graduate student, and Scott Retterer from Oak Ridge National Lab helps us fabricate these ZMWs. So what is this ZMW or zero mode waveguide? So as I said, it's basically a cover glass coated with aluminium, but with these holes that are 100 to 200 nanometers in width. I mean, we make them very precise, so we can make them 120, 160, 200, etc. And in fact, by controlling the size of these holes, you can change the excitation volume. So you can change the length constant of that evanescent wave by changing the size of those holes. So what the ZMW is typically used in a single molecule fluorescence microscope. And what this is basically a standard fluorescence microscope. We don't need to use TURF, although we have used TURF. And then this is connected to, you have this excitation light source, you have objective, you have the ZMWs, And then the light is collected by two cameras, a donor and an acceptor. One of the advantages of using this kind of a fabricated waveguide is that you can obtain data at very high throughput, right? And this is shown, we are showing a micrograph of one of these waveguides with each well containing a protein. And you can see these are many wells. So in principle, you can obtain large amounts of data using this method. So for a single molecule, when we started this experiments, we wanted to do what we thought were crucial experiments to or proof of principle experiments that established this concept. And so what we did was we took the cyclic nucleotide binding domain, we chopped off all the transmembrane regions that are there in the HCN. We just took the cyclic nucleotide binding domain, put a fluorescent tag on that domain so that we can watch it when it goes into the ZMW wells. And then we made these donor fluorescent ligands, and we looked at the ligand binding to this protein. And as shown here, you can see what we were looking at is FRET. We will excite the donor, and we are measuring emission from the acceptor. And this is what we are doing here. And in this case, you can see ligand binding at 100 nanomolar concentration. When the acceptor fluorophore bleaches, you don't see any FRET anymore. Okay. And you can go up to pretty high concentration. You can go up to 10 micromolar concentration, and you can see the number of binding events are much more, as you would expect as you go to higher concentrations. From that data, we could analyze from the bound events, and what we found was that the bound time distributions, we could generate these histograms of bound and unbound times. And what we found was both the bound and the unbound times, they fitted best to two exponentials or a bi-exponential rather than mono-exponential, suggesting there are two populations of bound state and two populations of unbound state. And we came up with this model, which was basically saying that when there's no ligand, the protein is fluctuating between two conformations, and once ligand binds, it undergoes a conformational rearrangement, and that kind of traps the bound ligand in that state. So once we did that, we wanted to take it to the next step, and so we wanted to do this systematically. The first I showed you, we did these measurements with monomeric CNBDs. Next, we wanted to establish this system with tetrameric CNBDs. And the reason we did this will become more apparent to you in the next few slides. But once we established this with the tetrameric CNBDs, then we wanted to go and do this with intact channel. Okay, so the thing is that once we started to look at ligand binding, the first set of experiments we did was with FRET between acceptor that is on the CNBD domain and the fluorescent ligand, which when it comes and binds to the protein, you see the FRET. But once we start doing multiple ligand binding, the FRET becomes very complicated, because you might imagine there might be the ligand might FRET to the donor that is sitting on its own subunit, but it might also FRET to the neighboring subunit. So we knew that we could not use the FRET technique to do this analysis. So the only way was to go back and basically then look at ligand binding itself. And the way we did that was to the original idea that as soon as the ligand binds here to this protein, you see a bound dwell time, and that corresponds to the bound ligand. So the problem here is that okay, so this is the expectation. The expectation is that as more ligand bind, you will see these jumps or these step jumps corresponding to ligand binding. Okay. But when we started doing these experiments, so we did these experiments. At that point, we made these tetramers. That was this tetrameric ion channel, a tetrameric subunit. And the way we made this was we took the CNBD domain, we attached a coiled domain on the C-terminal end. Excuse me, a coiled domain on the N-terminal end. This coiled domain forms a tetramer, and that's how we created the tetrameric CNBDs. So there is no HC at the transmembrane region. It's still the cytoplasmic domain, but all of them tagged together. Okay, so what we did was when we actually collected data, what we realized is the data looks really noisy, and there's the multi-state idealization is actually quite challenging. And it turns out that many of the algorithms we were using or available at that point were not good enough. They were, in fact, we know in this case, it's a defined system. We know there are only 4 ligand binding states, but when we use these single molecule algorithms that were available to us at that point, we found that in some cases it was fitting to 8 binding steps or 6 binding steps. So the data was being overfit by many of these algorithms. That said, many of these single molecule algorithms are designed for FRET measurements, not necessarily designed to do this kind of a single recording designed for these measurements, which are measuring binding and step jumps. So my graduate student at that time, David White, spent few years trying to develop better methods, which are, first of all, more accurate, and then are also faster for idealization of the data. And so this work, I'm not going to go into a lot of detail here, but the main thing that I want to note here that our method was much faster than the existing method. So as you can see, they were, in terms of the idealization, as the number of data points increases, our method was significantly faster. And this is important for us because we can generate a ton of data just because we are monitored many binding events at the same time, and we can get a lot of data. A typical single molecule experiment lasts a minute, whereas in our case, we can go up to 10, 15 minutes of data collection. And the main reason is the fluorophore, since it is binding in and going out, it doesn't bleach like in a typical single molecule experiment. So we generate a lot of data, and so our data is very well constrained. So what David found was that this method, which he called DISC, was much more accurate in terms of being able to idealize, as well as much faster. And then he used it to idealize his data traces and apply, and then extract information from there. And essentially, as this is showing you, we collected this data from this tetrameric CNBD. We collected 34.5 hours of data, and we from 293 molecules. So it's a lot of data which was analyzed. And what did we find? So one question that of relevance that was still there is that if you just take the cytoplasmic CNBDs and put them together, do you observe any cooperativity? Because if you do, that means that the cooperativity is at the level of cytoplasmic domain, and it doesn't have anything to do with HCN channel. And that's an important question that we thought our construct might be able to address. I mean, granted, this is a tetramerized CNBD. We are kind of artificially bringing them together, but we thought that this might be something that we worth exploring with these studies. So we did, from our data, we can get two sorts of information. We can get state occupancy. And if the idea is if it is non-cooperative, the occupancy of each ligand will follow a binomial distribution. So if each of the ligands bind independently, then the distribution, the occupancy will be a binomial distribution, right? And so the raw data looks like this for a typical concentration. I don't remember what concentration we used here, but the point is when you take the binomial expected by distribution from a binomial fit, you find that the data actually fits very well. So it looks like the ligand binding to the tetrameric CNBD is quite independent. Now, the other way we can do is to look at the rate, the transition rates, right? Now, for a independent model, these transitions will be quite independent, or will be basically there will be a statistical factor in front, but the K on and K off is will be the same for the first ligand, second ligand, third ligand, and fourth ligand, right? And essentially, this is what we see. We see a linear dependence, and this linear dependence correlates with the statistical factor that you expect here. So these are our values, and and again, in this case, it doesn't appear that the binding is cooperative. So the tetramer CNBD behaves as if independent of each other. So in that respect, maybe this is not surprising, but for us, it shows the most important thing is that it shows that we can use these binding measurements to determine the state occupancy and the transition rates, which ultimately inform us about the cooperativity. So at this point, we thought that we were ready to take up the next question, is whether the full-length channels, the HCN1 and HCN2 channels, exhibit any cooperativity. And this work was done by David and Sandeepan. Sandeepan came to the lab. He knew how to do the biochemistry with the HCN1 and HCN2 channels, and he set up all of that in the lab for us to do. And what these, and the reason we took HCN1 and HCN2 is HCN1 at that point was the one whose structure was available. We don't have a structure of HCN2, but all the studies that I was showing you at the initially for non-monotonic cooperativity that have been published in the literature, that comes from HCN2. So HCN2 is really the molecule that is thought to exhibit non-monotonic cooperativity. So we collected these data from both HCN1 and HCN2, and these can be idealized. The data looks quite reasonable. We collected a ton of data, as I was mentioning. We collected 60 hours of HCN1 and 35 hours of HCN2 data. And all of that data was used to analyze using disk, as I will show. So what are the things that we learned? First of all, when I told you about the monomeric CNBD, remember I said there were two unbound states and one bound state, two unbound states and two bound states based on the bi-exponential distributions of the bound time histograms and unbound time histograms. So when we looked at the HCN1 and HCN2, so one of the things we noticed is that in this case, most of the bound time distributions are bi-exponential, whereas the unbound time distributions were mostly mono-exponentials. And in this case, this is HCN1, this is HCN2. Both of them, the bound time distributions are bi-exponential. And you can see an example here. You can see this short dwell times, and then this example where you have the long dwell times. Okay. So that means that in this case, we are not seeing the unbound conformation to be that flexible as we had seen when we took the monomeric CNV. So now what happens to the ligand binding? And this is the key experiment that we set ourselves to do, right? When we looked at the state occupancy, HCN1 and HCN2 over various concentrations, and in gray lines there is data, state occupancy from various molecules that we looked at. So each individual molecule, we plotted the state occupancy. What you can see is that the red line kind of really fits well to the state occupancy that we observe. And the red line corresponds to a binomial fit. And that distribution fits over all the concentration range for HCN1 as well as for HCN2. So that itself told us this, that the binding in this case seems completely independent of each other. We also did we also extracted the transition rates using this simple sequential model. And again, with HCN1 and HCN2, the transition rates don't seem to be, they have linear dependence, and the linear dependence match the predicted one. And there again doesn't seem to be any cooperativity. So the previous data or previous work had shown that there was this non-monotonic cooperativity. When we purified these channels and looked at cooperativity at a single molecule level, we don't observe anything. What we could do was, given all this data, we can use it to try to understand how HCN1 and HCN2 differ and why they differ. What we found was that if you take this data and fit it to a model, and the model is the following, that the ligand binds first to one conformation, but then this molecule undergoes another conformational switch, okay, and this is the second bound dwell time. Then there are 4 subunits, so that's why you get this kind of a model. And you can estimate what are these transition rates for each of these transitions. And what it seems that the main difference between HCN1 and HCN2 is that the HCN2 is able to make this switch to the second state much more effectively than HCN1. And perhaps that's one of the reasons why HCN1 shows much less voltage dependent shifts than HCN2. Okay, so most of this work was done a couple of years ago. And since then, we have been trying to push this and ask the question, what happens when you put these channels in digitonin or in lipid? And the goal was to understand if lipids play any role in regulating membrane, in regulating ligand binding, etc. And the way we did this experiment was, we first, all the experiments that I showed you before this was HCN channels purified from membrane, but then solubilized in detergents. The detergents that we solubilized them were the same detergents where the cryo-EM structures were solved. But then here we took the next step to ask, now what happens if you reconstitute them and put them in a more lipid native environment? So what we did was we reconstituted them in MSP nanodiscs. Okay, so these are nanodiscs, are proteins, are membrane scaffolds. Essentially, you have a piece of membrane that's surrounded by these MSP proteins, and these, which are called membrane scaffold proteins. And then you can have your HCN channel or any transmembrane protein sitting in the center. And so this provides a more native-like environment for the protein, more, and it has this lipid. So what we did was, so initially HCN channels in digitonin, that's the data that is shown here. when we put HCN channels in nanodisk in lipid, soy polar lipids, the data is shown here. So what is you will see is interesting here already in these kind of raw traces is that we observe many more bound states than we observed in the in presence of digitonin when the HCN channel. In fact, one of the things that we observed is that when you plot expected to observed ratios, that's plotted here, it should all, if it is a binomial, if it fits according to binomial distribution, they should all be on this straight, on this red line. Okay? But you can see that there's a deviation from this for at low concentrations at 100 nanomolar. This is something that we have started to look at much more carefully and try to understand where this is coming from. And what it seems like, and I don't have the time to go into this today, but what it seems like is that in HCN1, if you take this data and generate a binding curve, what you see is that the HCN, the ligand binding seems to be slightly more cooperative in presence of nanodisk than in presence of detergent. This is somewhat surprising result, and yet still trying to make sense of it. But this is one of the most interesting points is that for me is that now you have ligand binding to a cytosolic domain, but somehow it has is influenced by lipids, which are there only in the transmembrane domain. With that, I'm going to start to wind up here. So summary and future direction. So the main thing that I want to you to take away from this talk is that the distribution of the various liganded states can be resolved using single molecule binding measurements, and these are necessary to discriminate between various models of cooperativity. Second is I showed you how zero modes waveguides can enable measurements of single molecule ligand binding dynamics at micromolar concentration. Although I've not shown you how it can be done at millimolar concentration. This is something we have published, but I just didn't have the time to show some of that work. So contrary to existing models, we find that cyclic AMP binding to detergent solubilized HCN1 and HCN2 channels occurs completely independently in absence of membrane potential. And finally, binding of cyclic AMP is followed by a conformational flip in each subunit. The efficacy of ligand-dependent activation is related to the stability of the flip state. And some of the questions that we are trying to address is cyclic AMP binding non-cooperative or in presence of membrane lipids. I would say our initial data or preliminary data looks like that there is some cooperativity in presence of membrane lipids. Is cyclic AMP binding different? How does the cooperativity, or if there is cooperativity when the pore is open at hyperpolarized potentials? How do allosteric antagonists modulate cyclic AMP binding? Those are some of the questions that we would like to answer in the future. With that, I would like to thank the people who are involved in this work. David White, who was a graduate student in my lab, he did most of the, he developed the software as well as did most of the work on the full-length channels. Sandeepan Chaudhary did the work also with David. Marcel started the work in my lab, and Dima was involved in some of the structural biology that we worked on in this project, and he helped Marcel with some of the biochemistry. Vinay and Susavan are the two postdocs who are currently working on this, trying to understand how voltage as well as lipids change the ligand binding cooperativity. I would also like to thank my collaborators, Randy Goldsmith, Scott Ritter, and the funding agencies who have supported this work. Thank you for listening.
Thank you very much to you. Very nice work.
Thank you.
So let me go through some questions. Eduardo Gomez, who is in charge actually of the seminar, is asking, what is the relevance of cooperativity to the ion channel's function?
So one of the recent studies show that, so first of all, we don't know whether there is cooperativity. If you look at the binding curves, they have a Hill coefficient of 1.2, 1.3. So it suggests there is some cooperativity. It's not absolutely cooperative. It's not one. But one of the things that it turns out is the question about how many ligands are necessary to open the channel. And that's also not a straightforward question. It seems like right now, not based on our work, based on work that's out there, is that ligand binding, when it is, when the channel is closed, the ligand binding seems to have binding of each ligand has a linear effect on the channel opening probability. But if you drive the channel towards opening, binding of two ligands is sufficient to fully open the channel. So the extent of cooperativity that you see depends on what state the channel is, whether the channel is closed or open. And I don't really have the full sense of how this is affecting the cooperativity is affecting the channel, because we haven't made a channel that is, first of all, non-cooperative to us in a physiological context, what would happen to that channel. But also the other point is that the cooperativity seems to be dependent on whether the channel is closed versus being open. And that's also the other question. So it's not as clear-cut as, for instance, the kind of answer that somebody studying hemoglobin will be able to give you. I don't know exactly. But my motivation for studying this was this unusual non-monotonic cooperativity, that was suggested. This was the established model in the field for many years. And the question is, how do you have a tetrameric channel which exhibits non-monotonic cooperativity? So this is something I thought would be.
Okay. The next question is from Elena Lopez. Does the independent binding of cycle AMP can be corroborated with microscopic recording of HCN channels?
So the microscopic single channel measurements for HCN channels have been difficult because they have one femtosiemen, one picosiemen single channel conductances, so which are harder. So not too many labs have described that. Secondly, it is really hard to show that how many ligands are bound when you are looking at this activation. And one experiment, I believe, that was done many years ago, early 2000s, where someone had looked at CNG channels, where they had used ligands that are basically one ligand, the two ligands which were tethered to each other. So the idea was that when one ligand binds, then it increases the binding probability of the other ligand, and the tether length was adjusted that the ligand binding would be that two ligands would bind at the same time. And again, that experiment seemed to suggest cooperativity, but it's always hard to know when a ligand has fallen off from one of the binding sites. I think that experiment has not been done. I mean, ideally, I would like to do an experiment where you take a channel and you knock off 3 of the binding sites and only work on one binding site, and then ask that question. That experiment remains to be
Okay. So the next question is from Physiology USLP. How are the changes in membrane potential communicated to the cyclonucleotide binding domains?
Yeah, that's a good question, because so we think that the changes in the membrane potential is not directly communicated. We think the main effect is that the membrane potential drives channel opening, like when you make the membrane more hyperpolarized, that favors channel opening, and the pore is connected to the CNBD, and that changes how the CNBD or the C-linker part of the CNBD domain is interacting, and that's what is affecting the cooperativity. But we don't have evidence for that. This is just my hypothesis based on the structure. I don't think there is any way membrane potential can directly affect cooperativity.
So just in the same direction. So you think that if this ZMW measurement were done at minus what 130 millivolts, you will get the same non-cooperativity?
Well, that remains to be seen. I would say that remains to be seen. We don't but it could be. It is quite possible, and I think that's because if you open the pore, the way this, if you look at the structures, the S from the pore helix to the S to the C-linker. C-linker is the region that connects the pore helix to the CNBD domain. The C-linker, it's very tight connections, and I think the position of the pore helix changes how the C-linker is placed to each other. And based on our understanding of the structure, I think the C-linker is the one that mediates, if there is any.
Yeah, okay. So I am assuming that these binding sites are identical, right? So each one
They are.
Unit. So had you tried to make a concatamer where you only have one binding site active, 2 or 3?
Yeah, we have thought about this. We haven't done that experiment. People have already made that concatamer and it works, so we know this would work. But that is one of those experiments, because if we see cooperativity, we want to ask how
exactly.
This is. Yeah, exactly. No, this is the plan right now.
So in the experiment, in the CMW experiment, so you was using one monomer, right? And then in the second step, you use the 4 monomers together. So I was wondering if in using the same idea, try to have one active site and the other one mutated. So how this measurement will be resulted? Are they going to be altered by not having a active binding site, or?
Are you talking of in the tetrameric CNBDs, or are you talking of in the full length HCN?
In the tetramer.
I wouldn't think it should be affected, but we haven't done that experiment. I don't know why it should be affected, because we don't see in the tetramer, the problem with the main purpose of the tetramer for us was whether if we see cooperativity, then we would have said, okay, this is happening because the CNBD domains are able to interact.
Yeah.
But if you don't see a cooperativity, you can always argue that the way these are attached is not what is physiological. So maybe that they would never see a cooperativity. And we didn't solve the structures of those, so we don't really know. But so if there is no cooperativity, I don't see any reason why mutation of one should affect another one.
Okay.
I did not see a motivation for trying figuring it out. So
my last question is it's about the effect of the lipids.
yeah,
It seems to me quite interesting that having that they change the slope of the relationship between the activity and that at the end you were saying that probably in this condition there is more cooperativity and I was trying to rationalize how the lipids will do that.
That is it. I have only hypothesis and speculation, and again, this is a work that is we are working on. We are writing, but the effect is actually quite clear cut. We have data from multiple lipid conditions. We see this in HCN1 channels. We see in HCN4 channels. We see it in various conditions, and the idea is that I think that in absence of lipids, perhaps even though the structure looks fine, I think the protein is just a more dynamic in detergent. And so if it is what is happening in other is probably not transmitted as well. I mean, there's just more flexibility in the, and so the protein, if there is a conformational change in one subunit due to ligand binding, if the protein can adjust without any restriction of the lipid, maybe that means that the neighboring subunits don't feel the need to adjust, right? I mean, whereas the lipid holds it together in the nanodisk, then maybe that forces the neighboring subunits to adjust more. I'm not sure. This is just pure speculation.
So do you have any idea what lipid in a specific can be done?
We tried, we have, I don't have the data in front of me, but we tried various lipids. In fact, what it seems like just our main conclusion is that just having lipids is much more important than just going into detergent. It was not any specific lipid it was not like a lipid interaction that was determining the cooperativity. It's just having them in lipids was enough.
Okay, so there are a couple more questions here. One is, can negative cooperativity be due to a steric hindrance in the channels, in this channel?
No, definitely. There could be reasons to believe that there could be negative cooperativity too, and it would. I think the main thing is whether it is positive or negative cooperativity. We need to get a good measurement that tells us what is the cooperativity and what extent of cooperativity is. Only then we can understand when we make, for instance, mutation or disrupt something, how is that contributing to the cooperativity? The problem, as I was saying, that just from macroscopic data, that I don't think this is enough, would be enough, given all the issues that I talked about.
Okay. So there is one question that is not very clear to me, but anyway, it says, what is the effect of zero mode waveguides in the experiment? So I think, well, I don't know what.
Yeah, I see what this person probably is implying is that whether we see things differently if we do the experiment is the zero, because it is a surface. There is aluminum, there is all of that. And an argument or a question can be raised whether you are seeing something that is different from zero mode waveguide than in macroscopic. I think that is quite hard to completely rule out. In part because we can't do the exact experiment that we are doing in zero mode waveguide without them. But we can look at certain things. We can look at binding of, others have looked at binding of molecules which bind at much higher affinity, right? Inside zero mode waveguides and in using turf microscopy, and they find that pretty similar. I don't think they know. But nevertheless, that doesn't rule out the question about what happens in our case. So the only thing I would add here is we have done these binding experiments, a macroscopic binding curve. And from single molecule experiments also, we can recreate a macroscopic binding curve, just like the one I showed you before, right? The binding curve that I showed you is a binding curve that is reconstituted from single molecule data. And those look very similar. So I think what we obtained from a cuvette versus what we obtained from ZMW, at least those data look similar. Now, if there are subtle differences, that's hard to say at this point. So yeah, I hope I think I'm addressing the right question, I hope.
Okay. So one last question on my side is, so you already basically map up how these 4 ligands bind to the channels. But then the next question is how the binding, once the ligand is already bound, how this is transmitted to the pore. So there must be that all the transmembrane domain become moving, mechanical stuff, etc. Are you expecting some kind of allosteric events on that last part of the opening of the pore?
So, yes, what I am thinking is right now that the effect is mainly transmitted by through the S6 helix, because S6 helix is the one, the pore helix is the one that is connected to the C-linker region. And that's what I'm thinking is the main pathway for connection. Nevertheless, I cannot rule out the possibility that other voltage sensing domain also has some interaction with this. Yeah, this is an important question, but part of the answer for that question, once we establish what the cooperativity is and whether there is or not, I think some of that will come from structural data. It just has to be there. And that structural data will then guide us back to going back and say disrupting that pathway for communication and then asking what happens to the cooperativity. So I think both have to come feed from each other. And at this point, what I've shown you is just on one side of this story.
All right. So I guess we are on time. So I really appreciate that you accept and give us this very illuminating seminar, Baron. It was very nice to meet you virtually, obviously.
Likewise.
Hopefully, in one of these days, you are really become a Mexican and coming to the country and visit us. That will be great.
Sure.
Thank you very much and have fun.